Hi, everyone. I am joined here today by Dr. David Systrom, the Director of Open Medicine Foundation's Ronald G. Tompkins Harvard ME CFS Collaboration. And I'm really trying to follow up with him today on his work on a muscle biopsy study, which we actually talked about in an interview that we shared over the winter. So welcome, Dr. Systrom.
Thank you very much, Danielle. I'm happy to return and uh, give everyone a bit of an update on the project.
Awesome. So, as a quick recap of what we talked about over the winter, Dr. Systrom is conducting a study that's looking at the molecular changes that occur with post-exertional malaise in people with ME/CFS and long COVID that also meet ME/CFS criteria. And we know from existing research that people with ME/CFS exhibit physiological differences when experiencing PEM, which can be measured through a 2-day cardiopulmonary exercise test. But this study is really trying to expand on that knowledge to understand what's happening on that second day at a molecular level and in the muscle itself. So to start us off today, could you give us a brief update on where the study stands now?
Sure. So we have human studies approval to study a total of 50 patients. 40 of those patients have some either ME/CFS by classic IOM criteria, or additionally, patients with long COVID who meet the IOM criteria for ME/CFS. So it's before and after largely 2020 with acquired symptoms. And an important part of that old IOM criteria is the presence of post-exertional malaise, which you just mentioned, Danielle. So the ultimate goal of this study is to better understand the muscle physiology and any role that the mitochondria may have in precipitating post-exertional malaise. So we have targeted 40 patients with some form of post-exertional malaise and an additional 10 patients with fatigue and various diseases such as heart failure or pulmonary hypertension, but no post-exertional malaise. And where we stand right now is we are about 2/3 of the way through the study. Um, and patients are tolerating dual thigh muscle biopsies quite well. Our interventional team is fabulous, keeps them comfortable. And what I'm going to discuss today in the way of preliminary results is the baseline muscle biopsy. So that's the one done on day one. It's before an exercise perturbation, which is designed to precipitate a little post-exertional malaise.
Awesome. Thank you so much. So as you mentioned, we're kind of doing this follow-up interview today because you have some really interesting preliminary data that we want to share. So can you give a short summary of what you're seeing from these baseline muscle biopsies so far?
Yeah. So the preliminary findings suggest that we have an acquired mitochondrial problem. It's likely not a genetic form. It's acquired, and the signals that precipitate it, I think, are under active investigation. We don't know. They're broadly speaking, we think there's often autoimmunity and associated inflammation, and that those things are bad for mitochondria. So that's a hypothesis. Um, but the baseline studies suggest that we have what's called a decrease in mitochondrial biomass. And maybe a poor person's term would be the number of mitochondria in the skeletal muscle. And this is very, very different from the genetic form of mitochondrial myopathy. So those would be patients, they're often identified in childhood and with strong family histories, and then a well-defined either nuclear or mitochondrial genetic defect. Our patients have really the opposite finding. Patients with ME and long COVID, they have a decrease in the total number of mitochondria, whereas the genetic forms, we know from the help of our collaborator Bob Navio at UCSD, that the opposite occurs in genetic forms. In genetic forms, there's a primary decrease in the function of the mitochondrion. The electron transport chain is dysfunctional, and as a result, there's an increase in biomass or an increase in the number of mitochondria. one could view that as a compensatory mechanism to help um, stoke the power plant in the muscle and preserve the ability to exercise. But what we've got in a preliminary way with almost 30 patients is the opposite. We've got a decrease in the number of mitochondria or the biomass. Within those categories of up or down biomass, we also have um, emerging data from Baylor about the electron transport chain function. And it appears that it can go one of two ways. It can be decreased in addition to the decreased biomass in ME and long COVID. And the various complexes 1 through 4, those are the ones we can see with a frozen muscle biopsy, can be decreased. And when that happens, what we know is there is impairment in systemic oxygen extraction at peak exercise. So that's from a related, but it's a clinical invasive cardiopulmonary exercise test. So we know from the exercise testing that there is a subset of patients, predominantly women, who have impaired oxygen extraction. It contributes to symptoms and a decrease in the so-called VO2 max. So when we look at the electron transport chain itself in ME and long COVID, some of the patients have this defect in function and a decrease in biomass. One could view that as a double whammy. Presumably, the two things contribute to worse symptoms. But there is also a subset of patients who appear to have a compensatory increase in electron transport chain function. So in a way, you could say the primary defect might be a decrease in mitochondrial number, different from genetic forms. And some patients are able to rev up the function of the mitochondrion in a compensatory fashion. So it's diametrically opposed to the genetic forms.
Awesome. Thank you. So maybe I'll try to put it in my own words and you can correct me if I get anything wrong here, but it sounds like what you're seeing is that people with ME/CFS have this reduced mitochondrial biomass in the muscle, even at baseline, which is the samples that we're looking at right now. And that roughly might correlate to reduced energy production or worse symptoms. And we don't yet know exactly what's causing this, but there's this hypothesis around it where the cells are receiving stress or danger signals in some sort from the body, and this is kind of their adaptation to those signals. Does that sound right?
That's right. Or I guess adaptation might imply, I agree with everything, Danielle. Adaptation might imply to some that it's a favorable response.
Mm,
Uh, so I think that what we're seeing in a preliminary fashion is that in ME and long COVID, that there is what appears to be a primary defect in the biogenesis of mitochondria, meaning the numbers are fewer.
yeah.
And, um, and then some patients additionally have decreased function, and they are the ones we really identify with poor oxygen extraction and a depressed VO2 max, and presumably worse symptoms. There's another subset, however, who maybe in response to the decreased biomass do increase their function of the individual mitochondria. And that I would view as a favorable adaptive
Yeah.
phenomenon.
That's a great nuance. Yeah. And I mean, speaking of nuances, I think, we talk about mitochondrial dysfunction in ME/CFS research a lot. And so it's really interesting. I think what is kind of fascinating about this preliminary data is that you're seeing some of the level down of that mitochondrial dysfunction with the electron chain transport, sometimes being defective in some of the mitochondria when you're seeing that in a physiological sense as well with the poor oxygen extraction. But then there's the subset that, like you're saying, is adapting better. So that's a really, really interesting finding, I think, that you're seeing.
Yeah, exactly. And I think maybe just taking one giant step backwards, what we've identified for a long time now is this issue of impaired oxygen extraction. So oxygenated red blood cells, for the most part, in both ME and long COVID, during incremental exercise and at peak exercise, they're just fine. They're going out the aorta, the big pipeline out to the exercising muscle. But then in a subset of patients, something additionally happens, and that is impaired oxygen uptake and utilization. And really, the differential diagnosis there is blood flow abnormalities at a microvascular level, so small blood vessels, and there are a lot of potential reasons for that. But the other possibility is that there is intrinsic mitochondrial dysfunction. And an important point for, I think, patients and docs to know, healthcare providers, is that the treatments for these things, at least currently, are totally different. Blood flow abnormalities on the one hand, and maybe related autonomic nervous system dysfunction, and then intrinsic mitochondrial dysfunction, either number or function. The current treatments are quite different. And ultimately, what we're hoping for, obviously, is precision medicine. And so we can see an individual patient and with our biomarker quest identify these subsets of patients and treat them properly, get them better.
What a wonderful way to wrap that up with some clinical relevance. I love that. So thank you so much for taking the time to provide an update on your research and share some really exciting results so far.
No, and thank you, Danielle, for asking the good questions and for helping organize all this.